This is a history of molecular biology, and let's jump into what really happened in history. Friedrich Meichner was a Swiss biologist and physician in the 1970s when he first isolated DNA from a white blood cell. His question is that really the DNA that's responsible for passing on heredity? That was the question at the beginning of the 1900s. In the 1900s, scientists realized that the chromosomes contain DNA as well as protein. So their question was, what is really the chemical composition that carries heredity? So DNA or protein? The protein seemed to be a better choice. It's 20 amino acids make up different proteins, and this 20 can give a huge variety, can lead to the variety. The other one is the DNA, and the problem with the DNA accepting is that a hereditary material is that it contains only 4 nucleotides, so it seemed way too simple to carry out great differences as we see in heredity. So the studying of heredity in a molecular way would lead to molecular biology in the 1900s. Third point, we are going to talk about Frederick Griffith, is a British scientist in the 1920 with his famous experiment in 1928. He was after World War I when he wanted to have a vaccine for pneumonia cases in Europe. The bacteria what he used, the Streptococcus pneumoniae, and I underline because it's genus species name. And there are, so World War I, he wanted to have a vaccine for pneumonia, case of bacterial pneumonia. So the experiment, what he did, he found that there are 2 types of Streptococcus pneumoniae bacteria. One of them is with a very thick wall. It contains a caspid made out of carbohydrates, and it looks like, in a culture, it looks like very smooth bacteria, and the capsid gives a protection for the bacteria, and it's a disease-causing organism. On the other hand, the other types are the rough bacteria, or without any capsid, so no protection, and they don't cause disease. That was really kind of the 2 strains what he used in his experiment. So the first thing what he did, he got the smooth bacteria, the pathogenic one, put them into a syringe and injected the mouse with that bacteria. And what he noticed that these are the smooth bacteria, the Streptococcus pneumoniae. And what happened? The mouse died. Well, there's no wonder, that was a pathogenic bacteria. So, I mean, it was expected to happen like that. The second experiment, what he did, he got the syringe, got rough bacteria, the bacteria that doesn't cause disease, into the syringe, injected the content into a mouse. No surprise, the mouse was alive after the injection, even after a couple of days after. The third time, what he did, he got smooth bacteria into the syringe, the pathogenic, but before he placed those bacteria into the syringe, he killed them with heat. So they were dead bacteria, so the mouse, after injection, did not die, No surprise. What really surprised him was the fourth part of the experiment got those bacteria, the killed bacteria with heat, and mixed with rough bacteria that didn't cause disease or the non-pathogenic, injected into the mouth, and what happened with the mouse? The mouse died. He was really shocked, and I bet the whole science community at that time, because number 4 was kind of so hard to explain. So we know now, at this point, what was shocking, that the dead bacteria somehow made the non-pathogenic bacteria to be pathogenic bacteria. What we know now, that I should have pushed up, yeah. DNA was not destroyed by the heat, and the DNA material from the pathogenic bacteria was picked up by the non-pathogenic bacteria. So we call it now, this transfer is kind of a transformation, genetic transformation. So living organism incorporates extracellular DNA into their own genetic material. Now, that's what we know now. At that time, in the early 1900s, the question came up, how did the non-pathogenic bacteria turn into pathogenic bacteria? Came Oswald Avery, a Canadian-born American scientist in the 1944. Think about almost 20 years later, a little bit less, 16, that he proved that the DNA was the transforming agent in Griffith experiment. What he did, got the capsulated bacteria, used heat to kill them, and started to separate the bacterial components into proteins, and put the protein into a test tube, separated the carbohydrates, separated the lipids, separated the DNA and the RNA as well. So what he did, break down everything with the enzymes except the protein, so the protein was not broken down. For the carbohydrates, break down everything except the carbohydrates, but these are minor stuff. And what he did, he added non-pathogenic bacteria in each test tube. So he wanted to see what is the material that the non-pathogenic bacteria will pick up. So he had the mice for all of them, for all of the components, and he injected the mice with the protein, with all of the materials. All of them were really happy, except whatever got the DNA from the capsulated bacteria and the non-pathogenic bacteria died. So he pretty much nailed that the DNA is the transforming agent, but you know, in a science community has to be proven more than one way. So Alfred Hershey and Martha Chase, American scientists in 1952, came up with another experiment, and they proved that DNA is the transforming agent. What they did, what they knew at that time, the bacteriophage is a virus, and it's shortly called the phage, the T2, infects bacteria, and the virus reprograms the bacteria and makes the bacteria produce more phages. So they also knew that the phages contain only DNA and protein. So the virus makes the bacteria produce more virus. So only protein and DNA. So what they did, they got the beaker and they placed T2s into the beaker, as well as radioactive sulfur into the beaker. Sulfur is only used for protein. You can see sulfur only in proteins. So the T2 picked up the sulfur into its protein components, the radioactive one, radioactive sulfur. The other beaker contained T2 phages as well as phosphorus, radioactive phosphorus, and the phosphorus, we can find only phosphorus in the DNA. So what they did, they added E. coli to both of the beakers, and what they found out, the infected E. coli contained only radioactive phosphorus but no radioactive sulfur. So this really proved that the DNA was really the transforming agent that got into the virus and modified got into the bacteria and modified the bacteria to produce more phages. Now the last one is the virus replication, the phage. Let's go with that, the phage that infects prokaryotes, because the eukaryotes will be a little bit different. So we have the phage, and it's going to infect a bacteria, a host. The DNA gets into the bacteria but not the protein, and the bacteria is forced to make tiny phages, small bacteria, and small virus, and the virus, as it bursts out from the bacteria, will destroy the bacteria, the host cell.